Namaskar. Today we'll study insulin. We know that diabetes is a disorder in which there is either deficiency of insulin or insulin resistance, which results in hyperglycemia, and accordingly we can have two types of diabetes, type 1 and type 2 diabetes, where type 1 is the insulin dependent diabetes mellitus and the type 2 is the non-insulin dependent diabetes mellitus. And type 1 insulin type 1 diabetes mellitus is treated by insulin only, because you can see here normally there are two phases of insulin secretion in response to glucose or any other secretagogues. So the first phase is the release of the stored insulin, followed by the release of the synthesized insulin. But in type 1 diabetes mellitus, there is absolute deficiency of insulin, so you call it as insulin dependent diabetes mellitus. And type 2, the phase 1 is absent, only phase 2 is present. The body can synthesize insulin in response to drugs like insulin releasers, and it is called non-insulin dependent diabetes mellitus. So insulin is used for treatment of type 1 diabetes mellitus, and in case of insulin resistance we can use type 2, we can use insulin for treatment of type 2 diabetes mellitus. And if we see the classification of insulin, source wise we can classify them as beef insulin, pork insulin, or human insulin, which is prepared by DNA recombinant technology. And based on the duration of action, we can classify insulin into ultra short acting, short acting, intermediate acting, long acting. If the duration of action is 3 to 5 hours, it is ultra short acting, and if it is 5 to 8 hours, it is short acting, and if it is 8 to 12 hours, it is intermediate acting, and almost 24 hours we call it as long acting. So lispro-insulin, insulin aspart, these are examples of ultra short acting insulin. Regular insulin, which is soluble crystalline zinc insulin, it is an example of short acting insulin. And NPH insulin, that is neutral protamine H-down insulin, that is intermediate acting. Lente insulin is also intermediate acting. Ultra lente insulin, insulin glargine, these are examples of long acting insulin. We can have also pre-mixed insulin, where we can combine regular and NPH insulin in the ratio of 30-70 or 50-50 or 25-75. So insulin, how it acts, the mechanism of action of insulin. Insulin receptor is a tyrosine kinase linked receptor, tyrosine kinase linked receptor. And as we know, this tyrosine kinase linked receptor follows two pathways, Ras Raf and JAK-STAT pathway, and here insulin follows the Ras Raf pathway. And this insulin receptor functions when two insulin receptors come together to form this heterotetramer. This is the beta subunit and this is the alpha subunit. So beta alpha alpha beta heterotetramer form has to be there. Two insulin molecules will bind to these two receptors, and there will be autophosphorylation of this tyrosine kinase unit. So this beta subunit is nothing but the tyrosine kinase unit, which will be autophosphorylated when two insulin molecules will bind, and this phosphate will be transferred to RAS, which in its resting state will be linked to guanosine diphosphate, GDP, and this phosphate is transferred to RAS by means of GRB2, and then this GDP will be phosphorylated to GTP. That means RAS will be activated. now this Ras GTP can transfer 1-phosphate to Raf, which will follow a cascade of enzymes. So RAF will phosphorylate MEC will phosphorylate MAPK, mitogen activated protein kinase. So MEK is MAP kinase, and RAF is MAP kinase. So this MAPK will phosphorylate various transcription factors which will regulate the gene transcription. So insulin regulates the gene transcription of a number of genes, and what it does, it causes the synthesis and recruitment of GLUT. So on the cell membrane, glucose, as you know, this is water soluble molecule, so it requires a transporter. We call it as GLUT or glucose transporter. So glucose will bind to this GLUT, and then this GLUT will transfer this glucose into the cell. So this is glucose molecule. So this is a protein, so carrier protein. So this synthesis and recruitment of this GLUT on the cell membrane is done by insulin. So the genes for GLUT is gene transcription of this GLUT will be regulated by insulin. Then there are mechanisms by which this glucose level can decrease. Glucose is stored as glycogen in liver and muscles. So the process of conversion of glucose to glycogen is glycogenesis, and the glycogenesis requires a number of steps by enzymes. So enzymes are required, and the enzymes are nothing but proteins. So all those proteins, protein synthesis requires gene transcription. So insulin increases glycogenesis by regulating the transcription of genes involved in the enzymes required for glycogenesis. Similarly, glycogenolysis is the process by which glycogen breaks down to glucose. So this is inhibited. So it will this insulin decreases or inhibits the gene transcription of enzymes involved in glycogenolysis. Similarly, gluconeogenesis is a process by which from non-carbohydrate sources glucose is synthesized. Non-carbohydrate sources like amino acid, fatty acid are converted to glucose by the process of gluconeogenesis. Again, this process also requires enzymes, and insulin inhibits the regulation of gene transcription, inhibits the gene transcription of enzymes involved in gluconeogenesis. Similarly, in the adipose tissue, insulin inhibits lipolysis and favors the synthesis of triglycerides. So in the adipose tissue, the fatty acids are converted to triglyceride and inhibits, so this stimulates the lipogenesis, whereas inhibits lipolysis, that is conversion of triglyceride to free fatty acid. So this is inhibited, and free fatty acid to triglyceride synthesis, that is lipogenesis, is stimulated. And it also increases the amino acid entry into muscles, and there favors their synthesis to proteins, and it stimulates the vascular endothelial lipoprotein lipase, thereby increasing the clearance of VLDL and chylomicron. So this is one benefit with insulin. So these are all actions of insulin. So we can use insulin in treatment of type 1 diabetes mellitus, and insulin is the drug of choice in case of gestational diabetes, that is diabetes during pregnancy. And in type 2 diabetes mellitus, as we have already discussed, that type 2 diabetes mellitus can be caused by either relative deficiency of insulin or insulin resistance. When there is insulin resistance, we can combine insulin with oral hypoglycemic agents. And the drawback with insulin is it can cause hypoglycemia. So this is all about insulin. Thank you.